Good afternoon, everybody. Just two housekeeping notes before we begin. First of all, the air conditioning for this venue can't be adjusted. It involves a trip out from Belleville in a bakkie by a man who unlocks a safe. So we can't just turn something down. Also, there have been some complaints that the sound in this venue is a little echoey, but this is as good as it gets. It's my pleasure to introduce to you this afternoon Professor Ambrose Wonkam. He's a senior specialist in the Division of Human Genetics here in the Faculty of Health Sciences. In addition to his clinical activities as consultant medical geneticist, he teaches registrars as well as genetic counselors and genetic laboratory staff. After his initial training at the Faculty of Medicine and Biomedical Sciences in the University of Yaoundé 1 in Cameroon, Dr. Wonkam completed a thesis in cell biology at the University of Geneva, for which he was recognized with the 2003 Denver Pinard Prize. Professor Wonkam has more than 30 peer-reviewed publications and 2 chapters in the book Genomics and Health in the Developing World, published by Oxford University Press. Welcome, Professor Wonkam. Thank you.
Thank you. So, bonjour. Bon après-midi. Oh, fantastique. Je vais très bien, vous? Oh, someone is complaining too much French, so we'll probably have to switch to English. But I warn you, it's only my third spoken language, so you have to adapt with a broken African French accent. I'll be able to speak slowly so that we can understand each other. So, first of all, my best wishes for 2020. Some of you may know that 2020 means vin en vin in French. That means that this year we're going to have 2 glasses of wine all the time. Vin en vin. The second is that I'm very grateful that you selected this specific lecture this afternoon. And I will try to convince you that as a geneticist, genetic science as a whole is important for the global society, beyond medical science and beyond ethics that go with genetics. Let's start with the beginning. Genetics, of course, have always existed, but the modern genetics as we know today was described by this man, the monk, Dr. Mendel, and he studied and he shows evidence of the mathematics behind heredity. A second important step in genetics was described by these 2 gentlemen, Watson and Crick, and the report of the structure of the DNA about 60 years ago. Anecdote says Watson and Crick spent a lot of time in the bar of the university, not in their lab. So it might be very good to spend time in the bar. Le Jeune in France associated the presence of 3 chromosome 21 and Down syndrome. But Down syndrome was already known for some time, described by Dr. Down. That was a British pediatrician. And he knew the condition also very well because her granddaughter has Down syndrome. But when Chijo and Levan, Chijo was a Japanese student, Levan was a Spanish PI, described the number of chromosomes. A few years later, Le Jeune shows that because of the extra number 21, you can have Down syndrome. But this afternoon, I will spend most of my time to discuss with you what to us is the most dramatic changes in knowledge in genetics, which is the Human Genome Sequence Project. This project was said to be completed in 2003 by President Bill Clinton, but I think it was just a way to have a political legacy because we are still working on it. It's not yet finished. I will not be finished before the next 1,000 years. The project was designed in the United States initially by the Department of Energy with 5 main goals: to identify genes that we have in our DNA as human, to determine the sequence from chromosome 1 to chromosome 22, to store the information in databases, to transfer the information related to it to new technology, and also to address ethical questions associated with the study of genetics. How did they start? Everything starts with inspiration from Africa. Some of you, or most of you, know the Great Pyramid in Egypt, and some of you probably have visited the pyramid. Some of you know the Rosetta Stone that is in the British Museum, just to show that I wanted to be sure that I can see it myself. And none of you know that this is my uncle from France. And when this man called Jean-François Champollion visited Egypt in 1818, following Napoleon Bonaparte, he was the one to find this stone and to describe the script in these 3 languages. The first is Egyptian, the second is Demotic. Demotic was a kind of lay person Egyptian, and the third was Greek and Roman. Of course, he was a linguist. He was very familiar with Greek and Roman, and a little bit with Demotic because he spent a lot of time in Egypt. And by comparing the 3, he was able to provide Is that fine now? He was able to provide the secret behind the hieroglyph. And from that time, a new science started, the Egyptology. When people that wanted to describe the human genome, at the beginning they didn't know where to start. And someone said, let's just do like Jean-François Champollion, let's compare things. And the first level of comparison was the comparison within our genome. The second was the comparison between species. The third, the comparison between you and your neighbor. And the next, the comparison between cell state. For example, in an organ, like the liver, if there is a cancer, there are some cells that have cancer, some other cells that doesn't have cancer. What is the difference between their genome? So we will try to follow this step to see what they have described. The project started in 1990 and finished in 2003, at least the first draft. And what we found by comparing the genome within the genome is that surprisingly we have very few number of genes. We have about 3 billion magic letters, A, T, C, and G, and we have only 20,000 genes. So as complex as we are as humans, we have only 20,000 genes. And how are the genes organized? They are organized like a big library, the library of life. Like in any library, you have shelves and you have books. And the shelves, the geneticists call it chromosomes, and we have 23 pairs of chromosomes. And within the shelves, we have books. Of course, we have 20,000 pairs of books. And within the information in the books, in reality, only 2% of those information code for proteins. So only 2% of our genes are responsible for a defined function that we know of the whole genome. So the 90% of our whole genome, what are they for? We don't yet properly know. Part of this are just called repetitive sequence. In fact, the first geneticists call it junk DNA, just to say that they are not there for anything. But we know today that even within the junk, we have to separate in 2 parts, the garbage DNA and the junk DNA. You remember the garbage is the one that you throw outside your home on Monday or Tuesday for the municipal people to throw away, right? That one, you never see them. You don't even know where it goes. The junk are those that stay in your garage. And one of these days you may reuse them. And some people you can donate them. So the junk, in fact, in terms of DNA, some of those repetitive elements, are useful in terms of evolution, as useful in terms of function. So we are still discovering what those parts of DNA are. So if we were to show our genome here as a big map, I'd borrowed this slide from my mentor, Stylianos Antonorakis. 1 or 2% are there to code for protein, for function. About the same level are what my mentor described about 10, 15 years ago that he called conceived non-genic sequence. That means part of our genome that doesn't code for protein, but that are the same for many generations of beings. When I say of beings, it means that some of those conceived non-genic sequences are the very same in the DNA of human, of chimp, of mice, of elephant, of wallaby. So apparently, some of those conceived non-genic sequences may allow us to distinguish the evolution, just the evolution of beings. That was a fantastic discovery for which he won the Allen Prize this year, in fact, in October. Let's summarize a little bit the comparison within the genome. We have 23 pairs of chromosomes. We have 20,000 genes. Only 2% of our entire genome code for proteins. If we move to the comparison between species, the two levels of comparison, one question I always like to ask is that who is our best friend in the animal realm? Amongst animals, what is the best friend for humans? Chimp. A different answer.
Best friend.
Yeah, best friend, yes.
Dog.
Dogs, of course. Dogs is usually the common answer, because the dogs are really the animal that we have every single day. There are people that will not even live without dogs. But chimp is the second best answer, because people will tend to say we are 98% close to chimp. We are kind of very similar. But none of you are right. I will argue that our best friend are mice. Why mice? We are 95% similar genetically to mice. Surprising, right? Someone will say, why not chimp? Because they are 98% similar to us. I wouldn't select chimp. You put only one chimp in this room, we're going to run away, all of us, because he's going to catch up, beat us, throw us away. One pregnancy in chimp, 9 months, exactly like us. So that means that for us as biologists, experiment in chimps is very difficult. For mice that are 95% similar to us, one pregnancy in mice, 19 days, you have 10 mice. 19 days. You can trust me on this. I spent 3 years of my life with mice, professionally. So if you want to repeat an experiment to understand human function, mice are the best animal for it, and informed consent is not a problem in mice. And someone will say, why is that mice are mice and we are humans? And why we have very 95% similarity? The only difference is that humans have developed more capacity of evolution to make more protein. And if you make more protein, you make more function. In fact, you have even developed capacity to combine protein to create additional functions. So we have 3 times more functions than a common animal that would be more than 90% closer to us. So comparison between species, the bottom line message is that we are closer to some species than we thought. And the best example are mice. The third level of comparison was comparison between individuals. What is the difference between you and you, you and you? And very surprising, I'll give you the answer, we are 99.9% similar. But 99.9% means that we are 0.01% different. So that difference may lead to a specific phenotype, specific condition, like this kid with albinism, with a mom that have a relatively dark skin, this other kid with albinism, because of a single small changes in one specific book on one specific shelf, what we will call mutation. Like this other kid, instead of 46 chromosomes, there is 47, and that lead to Down syndrome. This other individual, because of a small changes on a gene on chromosome X, there is hemophilia, and so on. And this fellow that could be South African with familial hypercholesterolemia. So those 0.01% are important. They may protect us, they may make us sick, and they may make us very different even from our parents, for example, with albinism. The level of difference between you and the next to you may happen at the chromosomal level. So imagine again shelves. It might be because one section of the shelves have been swapped. It may be swapped within the same shelf or in between the shelves. So within the same shelf is called intrachromosomal. Between two different shelves is called interchromosomal. Some changes may happen only at the DNA level, so you are not able to see them in microscopy. It might be a series of sequence, for example here, that is present in one of the chromosomes but not present in the next one. For example, some of those changes can make you sick. For example, in this specific address, if you miss that specific section, you have a specific condition called Cri-du-Chat syndrome, and so on. Some of them will not make you sick, but will allow distinction between family members. And these are called short sequence repeat. So it's just CA, CA, CA 10 times, CA, CA, CA 14 times, CA, CA, CA 6 times. And if you are from the same family, you tend to have those CA, CA, CA the same number of times. And these tests are the one we use for filiation, to know if people belong to the same family. And it's also the very same we use, for example, for transplant. All those differences are part of the 0.01% difference. And the last difference is a change in a single letter. Remember, we have 4 magic letters, A, T, C, G, that combine in a specific way to make the magic of life. Sometimes changes may happen in only one single letter, and those ones are called single nucleotide polymorphism. So this is a little bit the summary of the changes that allow us to distinguish humans amongst themselves. Either as a single letter change, either many letters put together, either a big chunk that is visible of chromosome, or many copies of the same gene. Why are these changes important? These changes are important because they evolve with time. If we were to live 1,000, 2,000, 10,000 years, our genome may not be the same because sometimes they are subject to environmental pressure. For example, this condition called sickle cell anemia is more common in sub-Saharan Africa, in the Mediterranean area, in the Arab country, in India, than anywhere else in the world. For the simple reason that if you had that specific changes, you resist to malaria. So wherever you have malaria, you have sickle cell. And that have made that changes very, very common in that population. Some changes also allow us to resist. For example, if you have a deletion, this is a receptor of white blood cell. If you don't have those receptors from your mom and from your dad, you are HIV resistant. Because that receptor is like the key for HIV to get into the cell. If you don't have the key will not get in. And every single time I give this lecture, some people will say, why is that we don't put that mutation in everyone? I will come to that a little bit later. So the third level of comparison between individuals. Surprisingly, we are very, very, very close to each other, 99.9% similar, but the 0.01% make us different in disease, different in responses to disease, different in responses to the environment, and also maybe healthier. And the last level of comparison now of the human genome is the comparison between cell state. And the classical example is a cancer cell. By definition, cancer are genetic condition. Cancer simply are cells that become mad and replicate without stopping. And this happens because of two things: an imbalance between stop replicating genes or continue to replicate genes. And the stop replicating genes are called tumor suppressor. Please continue to replicate genes are called, I'll find a name later. I'll talk to that later. So, but between the tumor suppressor and the oncogenes are those that ask the cell to replicate indefinitely. If you have mutation in two of those, the cells start to replicate. But for each generation of cancer cell, they accumulate some other mutation to try to escape death or also to try to invade your other cells. So cancer cells start to be intelligent, create some other mutation to escape death, and create some other mutation to get into bloodstream and invade, like metastasis. And based on this knowledge, by sequencing the genome of cells, there is a specific device called, for example, MammaPrint, that is a piece of glass with specific DNA design that allow to distinguish which cancer, breast cancer specifically, would be more aggressive than the other one. And according to the profile of the expression or of the mutation, specific treatment are given. So these information are important for medical practice. But do we know enough? My answer is absolutely no. There's still a lot of knowledge that we don't know. We still don't know how is that from the DNA here, you can manufacture a protein and address this protein to the surface of a specific cell. I know at least one person or two that have spent their whole life, 50 years in research, knowing why is that this small vesicle moved from here to here. They are working on cytoskeleton. So the whole machinery of the cells, how it's operated, there are a lot of questions that we don't know. But based on the information of the Human Genome Project, we can already use genetic information for therapy, use genetic information for gene therapy, use genetic information to manipulate drugs. Let's speak to that a little bit. The project started here, 1990, and every single day, there's a new disease genes that is discovered. Every single day. That means that most medical professionals cannot cope with the path of new discovery of genes. And maybe even in the room, someone already having information that I don't have because I haven't read all the literature of today. Every single day there is a new disease that has been discovered. What that means? That means that a simple general practitioner cannot cope with the number of disease. And a new specialty has been created called medical geneticist. The reason why I earn money every day as a medical geneticist. And what do? Our only role is to take the genome, look for variation, interpret those variation, and give the information to patients. And this is a specialty for which we are not very well equipped in South Africa. In the whole South Africa, we have about 15 medical geneticists, and we are about 55 million. So that means there are many, many provinces and towns and hospitals that doesn't have medical geneticists. So the other day, my little girl, she's 10 years old, she asked me, Dad, what do you do? She said, but of course you know, I'm a medical geneticist. He said, yes, that's the point. I don't know what a medical geneticist does. I know what a pediatrician does. He sees us. I know what the gynae does. She sees mom. But what do you do? And then I show this picture to my daughter and say, imagine this later and imagine this picture. Can you match them? And quickly she matched and say, that's very easy. He said, that was exactly what I do every day. I'm a matchmaker. I look for genetic variation in the DNA, and I try to imagine what is the disease condition that is associated with that variation. And I explain that to patients. That's as simple as that. That's exactly what every single day of my life I do. And every single day of the life of any molecular geneticist is summarized like that. 4 magic letters of our genome. What does that mean in terms of clinical practice? Some magic letters happen like in these genes here and lead to a very specific condition that can move from that to son. And in fact, if you look at this picture, this gentleman here, his name is Snape. And Snape lived in the 3rd and 4th dynasty, and he was a script, he was also a priest, and he was responsible to the wardrobe of the pharaoh. If you look very well, the wife of Snape is not like they have 2 kids. That here tells us at least 2 messages. One, genetic conditions have always been there many, many thousand years ago. The second, maybe our brothers for 3,000 years ago was more tolerant than us, because I wouldn't imagine easily a big group, many society because of the and as you can see, we were beautiful, not nice at the time. That was 3,000 years ago. We have to remember that from time to time. In this specific book here, this is a Nigerian woman with a single letter change here, and we will try to look for those letters and explain to the family that there is a risk of cancer in the family. We also use that information for genetic diagnostic before birth. We use the same information to design specific drugs for people, what is called gene edited. Remember CCR5. You remove CCR5, the 2 copies, someone will never have HIV. And he gene edited a baby that doesn't have CCR5. A major ethical debate, because these gentlemen have manipulated our common human genome. That means that from generation to generation, those 2 girls, that they were twins, they will have more kids that will not have CCR5. That means that 1,000 years down the line, you have individuals on Earth that have 1 gene missing. What are the consequences of that? We don't yet know. The reason why is genius, because he succeeded to do that, but it's also scary because we don't know what are the long-term consequences of the human evolution and of our common DNA. But it's been done. It's been done. It's been proven that it was correct. It's happened. Even though this guy was very much punished for that. I think his license was retrieved. He cannot practice anymore. He cannot work anymore in the medical field. Using genetic information also helps us to know where we come from and where we migrate from and where we are going to. Okay? As we all know, we are all African. And maybe we live in a country where the first human evolved. And if you can look at it in 2 ways. One way is by looking at mitochondrial DNA. There is one say that says mom have energy. It's actually true. Our moms have energy. Because in the mitochondria, which is, I wanted to use a company name that I don't want to use. It's in fact the major energy source of our whole cell. And that mitochondria got a specific DNA that allows us to have the energy we have in our body. And that DNA, we receive it only from our mom, not from our dad at all. So it's a genetic composition that we have from our mom that our dad have not contributed to? Zero. So they just make noise, dads, including me. But the energy comes from the mom. So that means that if we imagine mutation and read back those mutations many, many years ago, we are able to trace back where our very, very first mom come from. Because in mitochondria DNA, mutation rates are constant. And you can measure it over time. And by doing that measurement over time, we know that Eve had to be African. The biblical Eve was African. The second way of looking for human evolution, you can also use Y chromosome to be fair to males and to men. In female, there are 2 X's. In men, there is 1 X and 1 Y. Normally, when cell divides, all the pairs of chromosomes are put together, number 1 with number 1, number 2 with number 2, number X and number X in female, but in male it will not happen because you have one that is very tall, that is X, another one that is Y, that is very small. So they will not pair together easily. That means that they are part of our chromosome Y that doesn't change from generation to generation. That's the reason, for example, we know Fulani in Africa is a back migration from Europe because they all have Y chromosomes that are Europeans. That also means that we can follow back changes on Y chromosome for thousands and thousands of years. And by doing that, once again, the first human being came from Africa. We are all African. So if we did not have this genetics material or this knowledge, it would have been extremely difficult to prove that. Because before that, we tended to use anthropologic information that is very, very, very difficult to interpret. Yesterday I was visiting a Mosul Bay. There is apparently a cave that was discovered at the Pinnacle Point. I'll invite you, if you visit Mosul Bay, please go there. Apparently, that was one of the places where scientists have shown very recently that the first human behavior started. So we are really at the cradle of humankind in this country. Genetic knowledge also will help us in agriculture, in livestock breeding, also in bioprocessing. But genetic knowledge, as we have shown before, with the editing of the very first baby, human baby, is information that can be extremely powerful but extremely dangerous. So we need to be watched. Geneticists need to be watched what they do, because this can lead to confidentiality and privacy that is broken. For example, if we do a simple blood test for blood count in each of you, the next day we do the same test, we might not see the very same blood count, because you may have had a little bit of drink, because you may have not drink. That means that your blood concentration will be different. But your genetic information will be the same today, tomorrow, the next day, and will be the same 50% for your kids, will be the same 50% in your brother. So it's an information that doesn't change and that have major implication not for you, but for the generation before you and the generation after you. The reason why in our clinic, our information is not stored at the central storage facility. We have a completely different storage facility, because anyone that consults your medical folder can find information that may be related to your whole family, and we don't want that. So confidentiality is very important. Fairness of the use of genetic information is something that is coming more and more. Stigmatization is an issue. You probably have heard recently that in some part of the world, people that have albinism have been killed because they potentially may have magical power. They just have the genetic changes. 'Cause 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 Genetic education for doctor and health professional is something that will have to be addressed. And this is a very old picture. On this picture, you can recognize maybe one person, is me here. But I was 17 years younger. But the most important person of this picture is him, Victor McKusick. Victor McKusick is the founding father of what we do today, which is called medical genetics. He was the one that really started to say, we need to have doctors that specializes in interpreting DNA. And he created these courses that have been there for 60 years. That was the first time I arrived there as a student. I went there last year as a lecturer this time. It was a very great privilege. And when I was there 17 years ago, I got this slide from Professor McKusick when he was predicted what would become genomic science in many, many decades from that time. And he was predicting that, for example, in 2010, you will have predictive genetic tests for 25 conditions. I think he was pessimistic. 2020, which is this year, he was predicting that we will have gene-based therapy for diabetes, for hypertension, and for cancer. We do have gene-based therapy for many cancers, and not yet for hypertension or diabetes, but we are working for that. But he said something that was critical, that resonated to me as African, that the worldwide inequity will remain and contribute to international tension because the access to technology will be unequitable. I've spoken about only 15 geneticists for 50 million people in this country, for example. But he did not anticipate that the revolt of inequitable access will start in the US. A couple of years ago, that was exactly about 10 years ago, there is a company that is still there. That company is called Myriad Genetics. Myriad Genetics patented variation in genes that are responsible for breast cancer, BRCA1 and 2. What does that mean? It means that every single time you test for that in a clinical setting, you pay a royalty to Myriad. That means that if you are not able to pay royalty to Myriad, you are not able to know if your family or you have a predisposition to have breast cancer. Maybe Angelina Jolie will never have none, for example, because the royalty have not been paid. And it started to be very painful for professionals to even imagine that that is possible. So there was a case court that was initially initiated by the American College of Medical Geneticists, Patient Support Group, American Society of Human Geneticists, and they brought Myriad to court. And they won the case. And this is the first page of the Supreme Court of the United States that says that no part of our human genome should be patented. But remember, this is the United States, right? And it's only one country out of many, many in the world. So what Myriad Genetics does, they move the patent to Australia. So genetic technology is very powerful, but we need equitable access. And the only way we can do it is to do what we are doing today, but at all the level of the society. And to make sure that we have access to the technology and design solutions for issues that are related to our society. This is another example where inequity happens. For example, to understand why is that in some family you have diabetes that Mendelize, that goes from generation to generation, and in some other family you don't have. To understand why is that you are from the same family, someone eats McDonald's every day with 1 liter of Coke, but he will not develop diabetes. The other one that do the gyms every single day and eat only carrots will still develop diabetes. And they have the same gene package. To know that, a specific type of study is performed called genome-wide association study. But if you look at the proportion of genome-wide association studies that are performed in the world, the majority are performed on population of European ancestry. And last year, for example, 2 years ago, only 20% are performing in population that are not European. Up to last year, people will say that's inequitable access. But I will tell you a little bit later that there is a scientific imperative to do it in all the population. It's no more inequitable access. I'll come to that a little bit later. This is one of the reasons why all the population have to be studied thoroughly. This paper was published last year, and there was about 1,000 Africans from the Caribbean whose genome was fully sequenced. And they realized that variation in the genome of those 1,000 people, 10% of them was missing in the reference genome. 10% missing in the reference genome. That means that what I use every single day when I sequence someone and compare his genome to the reference genome, if that person is African, there is 10% of that information that will not be matchable with everything that is available in the database. In fact, the first reference genome was the reference genome of a friend of mine, Francis Collins. That is the director of the NIH. And the second one was the genome of Watson, James Watson, the one that described. And among the reference genome, we have about 10 of them. But what would that mean? I will tell you what is the implication of this. This is another friend of mine, Charles Rotimi. He's director of Global Health Center at the NIH in the United States. His team described a couple of weeks ago by studying population from South Africa, from Ghana, from Nigeria. They discover these genes that is responsible for type 2 diabetes. But they realized that the changes in this gene that is responsible for type 2 diabetes is monomorphic in European and Asian. That means that even if you have 1,000 people from Europe and from Asia and you study them, you will never find that gene. It was the perfect example where diversity in genetic study is critical for us to understand how our common human genome works. Because once again, there are some changes that accumulate according to where humans stay for a very long time, pushed by environment. Another example to see beyond the genomics. This is what we call EXACT for exome aggregated database. And EXACT, remember we say 2% of our genome code for protein. Those 2% is called exomes. So those are part of our genome that are responsible for the production of functions. So every single time I'm looking for one pedigree in one specific family and I found a variant, I need to go and compare that variant to a database that is called EXACT. If the variant is not present in EXACT, my next question is that, is this a mutation or is just a polymorphism? So rarer the variant is in the EXACT, more my computer will tell me it is a mutation. It might be pathogenic. But if you look at the composition of EXACT database here, European population once again is the big majority, the most studied. African population only 10%. That means that more mutation that I will find will not be present in the EXACT database. And I will tell you why this is important in clinical practice. One of the things that I do every single day, I study genetic of hearing impairment in children. So I have a major project that is funded at the level of $1 million per year. So it's a lot of people. We have researchers in about 7 countries on the African continent and in Europe and also in the UK. So the reason why nearly all the agencies in the world have funded us to do this study, because we show a couple of years ago that if we need to look to know what are the novel genes that are responsible why a child is born deaf, you have to study African population. They didn't give me money for charity, no. It's not because it's just the way it has to be done. And the study started with this. When we study population in South Africa and Nigeria, we realized that the genes that are known, that we know are responsible for hearing loss, will explain only 4% in this population. And even if you do the same study among African Americans, it's only 26%. And if you look at the higher end, Middle Eastern population, 70%. That means that we will not be able to discover novel genes if we study only Europeans or Asians. And to show that concept, we selected family that Mendelize. In all these family, you have 1, 2, 3 people that have hearing impairment, so they were born deaf. And because they are from the same family, it's highly likely to be genetics. So we sequence all the 20,000 books and look what is the mutation in those books. And we found the mutation in 7 out of 10 families that was already known to be responsible for hearing loss. That means that in 3 families, we were likely to find a novel genes. In fact, we discovered a novel gene this year. But we also showed that most of the mutation that we found here was novel variant. The message is that if we need to complete the library of life of variant, let's include all the population, because we come from the same mom. And if we just study the brothers that are at the top there, the other brothers that are not being studied, we are not going to have the full information. One practical example of that. 2 years ago in the United States, there is a condition called hypertrophic cardiomyopathy. Hypertrophic cardiomyopathy can lead to sudden death in young athletes. For example, in 2003 in France, in the semi-final of the Confederation Cup, a Cameroonian player called Marc-Vivien Foé fall in the stadium and die on spot. He had hypertrophic cardiomyopathy. 5 years ago, a player, I think from Bolton, called Fabrice Muamba, also fell during the Premier League soccer match. He didn't die. He was lucky because the paramedic was on the spot. He also had hypertrophic cardiomyopathy. For this condition, there is at least 30 genes that are known. And it's critical to know those genes because to those fellows, you're going to give them some specific medication. Beta blocker, in some of them you can put an intracardiac device to shock the heart in case the heart stops beating. There was one of these genes that was tested in the American clinic for a very long time. And there was one variant that had been given to many, many people for a very long time to tell them that they need to be treated because they have a mutation that would lead to hypertrophic cardiomyopathy. Until someone studied that variant in African American and realized that 1 out of 3 have the same variant. But they were very healthy. So that means that that variant was just a polymorphism. The only reason why it was said it was a mutation, if you check for those variants in European and Asian, it's very rare, but while being very common in African American. And that was a mistake because if we had even a small proportion in the reference exome, in the library of variation from that population, we would have prevented this. And this was a big problem because the clinic have to call back many, many patients to say, sorry, it wasn't a mutation, we have to remove your intracardiac device. That's very embarrassing. The message here is that human genome is a common good of humanity. If we want to know how it operates, we want to do it the same way in most populations, and that's the only way we can have the key of how it operates. I would like to narrow this to the African context. This is the world map based on innovation and wealth creation, and this is based on publication or discoveries. And if you look at that world map, Africa disappeared. But there is still a little section of Africa here. That's South Africa, right? I would like to believe that that section exists because of people like me, right? Because we have published a few papers and then suddenly it appears there. But what I would like to insist here is that we have 300,000 years of human genome history. 300,000 years of human genome history. And that should be our bargaining power on international negotiation. I'm speaking science. If you don't do it in African population, you don't yet know the whole truth. It's the only message. And every single time we say that, believe me, every single time it works. So today, we try to gave a little bit some information about the Human Genome Project here, but there are still many, many layers to be built around the knowledge that is here. How does the variation in human genome relate to our biology? We started using it for health, but we do not yet have the full picture. We started